Hi everybody, Paul and I were doing the numbers for the SNP lab, and we want to make sure that we leave a couple weeks in the semester just in case we screw up. And in order to do that, we have to start soon. So I'm going to give you a crash course in the skills you need to be able to do this lab. You don't need to memorize anything here, just kind of watch it, keep an eye on what's going on, try to remember as much as you can, and we will go over this again in class. Okay, so the first thing I'm going to do is I'm going to say, hey, Paul, what kind of personal protective equipment do I need? And Paul is going to say, Paul's going to tell me, I can't just assume that I need gloves. I'm going to have to ask because I never know as a new person what's dangerous and what's not. So, Paul, what kind of personal protective equipment do I need? You need gloves, Luke. Okay, thanks, Paul. All right, so I've got my gloves on. Second thing I'm going to do, I'm going to make sure that my work area is clean. What I usually do is I put everything together on my lab notebook, carry it to where I'm going to work, and then I clear off an area in front of me. I'm going to go ahead and take Windex. Make sure that I have a clean working area to begin. Circular motion to get stuff off of there. Cool. All right, next thing I'm going to do is start setting up. So in order to set up, I'm going to need a few things. I'm going to need this, which is a 1.5 mil centrifuge tube, which looks a lot like another type of tube that I'm going to show you. So this is a collection tube, and you'll see the difference here in a second. So I'm going to take a collection tube out of this bag, but I'm going to do something. I never reach into the bag. What I'm going to actually do is shake a tube out, and that's because I just don't want to screw up anybody else's research. I don't know how important it's going to be if they're going to use these tubes in the future. I know my area is clean, so now I have a collection tube. So take a look at the difference between the two tubes. First, one has a round bottom, one has a pointed bottom, and then obviously one has a lid and the other one does not. Okay, so anytime I'm using test tubes of any sort, I'm going to make sure that I have a test tube rack. I don't ever want to leave test tubes on I don't ever want to leave test tubes on the table. All right, so I'm also going to make sure that I have, since I'm going to be using tips and a micro pipette I'm going to make sure that I have what I call a dump beaker. So this is just going to serve as my mini little trash can. I'm just going to stick that over there. Okay, so the first thing I'm going to do Okay, this is a 50 mil Falcon tube. It now has a mixture of the cells that were in my cheek and hint water in it, and that is what we're going to get our DNA from. When you're doing this lab, it's going to be really, really important to know where your DNA is. It was in my mouth. It is now in this tube. One of the major mistakes made is that people accidentally throw away their DNA. I've done this several times. Okay. So I'm going to stick this to the side, and then I am going to go ahead and take a disposable pipette. Okay, I can't get my disposable pipette open. So we're just gonna cheat. Okay, so I've got my disposable pipette. I'm going to take some of the solution. I'm going to put it in here. Oh, sorry, wrong tube. I'm going to put it in my 1.5 mil microcentrifuge tube. Fill it up to the top. I'm going to throw my pipette into the dump beaker, put my microcentrifuge tube down, close up my Falcon tube. All right, so why am I doing this stuff? Well, everything that I'm actually doing right now is not dangerous to me, but it can be dangerous to other people. So I want to make sure that I'm closing tubes and as much as possible. So now that I have my tube, I'm going to go ahead and take a Sharpie and I'm going to label my tube. And I'm going to label it D period 1. And then I'm going to put a big 5 on it, because when I asked, somebody told me that I was going to be sample number 5. Okay. Just as a warning, you want to make sure that you label the thing that is attached to the tube. So, for example, if I was using this Falcon tube, I would not label the cap. I would label the side of the tube. Obvious reasons. All right, great. So I'm going to go off and I'm going to go centrifuge this down, and then we're going to take a look at what comes out. Okay, great. I just got done centrifuging, and now I'm going to show you what I did. When I centrifuge, if I take one tube, I almost always take another tube with me to counterbalance. When the things are in the centrifuge, they're spinning really fast, so if there's something on one side, I've got to make sure there's something on the other side. So I just took a tube of water with me. Okay, this is what my sample looks like right now. And if you'll notice, that's kind of hard to do. Focus, focus, focus. Okay, great. So I'm going to focus this by doing this. All right, here we go. All right, now if you notice there, I have liquid in the tube up to about here, and then I have a little white thing down there. And if we're playing the game, where is my DNA? Then my DNA is in the cells in the white stuff now. So what I'm going to do is I'm going to take my tube with my pipette. Now I'm going to hold my tube at about a 45-degree angle at eye level, push down on my pipette so that I'm ready to suck up some liquid. I'm going to go ahead and suck it out. And then keep doing that until I have pretty much just the pellet left. So there's no liquid in there. You can see there's just a tiny bit, and that's quite all right. Okay. So this lab is now going to ask me to resuspend a solution. I'm not going to go through that just because you've already done pipetting, but quick reminder, always check the pipette for the volume that it's supposed to handle. This is a 200, so that means it can handle between 200 and 40. Okay, so I'm going to go ahead do my pipetting. All right, and to do my accurate pipetting, I'm going to hold my finger right about here on the pipette, and I'm going to count down 3 as I go in. Pull up, 1, 2, 3, and then put it into the tube, 1, 2, 3. Great, and then I'm going to use my dump beaker, and I'm going to put my tip in there. So the other thing that you're going to kind of do is you're going to assemble, well, Okay, the other thing you're going to need to do is use your DNA isolation tube. This is called the DNA Easy column. We generally call this a column. So I'm going to go ahead and try to open this, and hopefully that will go better than the pipette did. Usually there's a corner that I can use. Great. Okay. I'm going to avoid touching the top of the tube as much as possible. Sorry, the inside of the tube as much as possible. So I've got my tube open, and you can see it's composed of two parts. It's got the column here, and then a collection tube. And what we're going to do is I'm going to take I'm going to take some of my solution, and then I am going to go directly over the center of the tube and pipette down into the column. So if I were to do it, I'm going to try to do it with you watching with one hand. Cool. All right, dump my tip. Now I'm going to walk over to the centrifuge. And I'm going to follow the centrifugation step. After I use the tube for the first time, the DNA moves. The DNA has gone from my cells and into this white filter. Okay, so at this point, the liquid that's going through this thing becomes unimportant. So the liquid that I collect in this tube, I'm going to throw away. And it's going to be this that I'm going to want to keep until just about the last step, because again, my DNA is going to be in that white little filter. So basically, I'm going to go ahead, trap my DNA in there. I'm going to clean it by adding a little bit of detergent and then adding a little bit of alcohol. And then once it's clean, I'm going to go ahead and try to drop it out of there. And I'm sorry, just got a text. I'm going to try to drop it out of there using a elution buffer. And that's the thing you have to listen for. So the elution buffer is going to be the one that drops your DNA out of this filter, and you're going to want to put that in a labeled tube that you can store. Cool? That's pretty much everything. Sorry for babbling. I'm kind of tired. And hopefully we can pull this off. Oh yeah, don't take off your gloves until Paul or I say you can.